Okay, the person we're waiting for is walking in, so. Well, it's my great pleasure to introduce today's speaker, Lori Goodyear from Harvard, and she's the professor and also senior investigator, co-head of the Integrative Physiology and Metabolism section at Joslin Diabetes Center. And I've known Lori for years. She graduated with a bachelor's degree from Springfield College, and which is the birthplace of basketball and a very important place for physical education. So she is a true leader in physical education and pioneer in the molecular transducer of physical activity. I've known her for, guess how many years? A long time. I calculated 33 years.
Wow.
Yeah. Yeah. So we exercise physiologists endure. Yeah. We stay forever.
That's right.
Yeah. And she's very innovative in research, but also very solid in, fundamental research. So her early work in glucose metabolism, glucose transporter, and the signaling, junk pathway, AMPK pathway really laid the foundation for the whole field. And now she and I collaborated, and we are both on the steering committee for the Common Fund consortium called MODAPAC, Molecular Transducer of Physical Activity. And she published 200 papers, and many in JCI, diabetes, PNS, Nature Communication, cell metabolism. So very impactful. But as I said, her work is very innovative and always touch the most important aspect of physical activity and exercise. So she studied not only the signaling, but also impact of maternal exercise to your children and the grandchildren. And she's studying the impact of exercise on adipose tissue. And I know she published paper, exercise impact on the brain. So, and if anybody want to follow the exercise research, just follow her research. And because of her work, and she got numerous awards, I'm not going to count them, but there are 2 or 3 I would like to mention. One is Frank Booth Award. Frank Booth is my mentor and one of the pioneers in the molecular exercise research, very well respected. So she received the Frank Booth. So we are in a family, okay? And then for her work, she got an honorary doctorate degree from University of Copenhagen and got to sit right next to the Queen. And so really impactful work. But there was one award you did not know. And I had a meeting before the grandfather of exercise biochemistry passed away several years ago. I had a dinner with him in San Francisco, John Halozzi. And John asked me the question, who is the leader in the field? And instead of answering question, I said, who do you think? And she said, Lori.
Really?
So that's an award, yeah. And so again, and the exercise field is not too big, but it's really like a family. And we collaborated, we compete in a very healthy way. And so one of the things I would like to mention before I end this lengthy introduction is that she trained many people and many influential research scientists in different fields, different countries. And one of them is coming to FBII, Sarah Lazar. And so, so today, Lori's talk is about epigenetic mechanism. I'm sure she will touch some other things, mediating the benefit of effects of maternal exercise on offspring metabolic health. And without further ado, Lori.
Thank you. Well, thank you very much. That was quite an introduction. I appreciate it so much. And hearing from John, the John Halozzi Award, I'll put that on my resume now, because that is, meeting the Queen was pretty amazing and pretty fun, but this is more, that's probably a more important thing to hear than it is that I got to know exercise. That's right. So thank you very, very much. So today I'm going to talk about my title was something about that exercise was good for health. And so that allowed me to talk about whatever I wanted. But what I'm going to focus on is written here, epigenetic mechanisms mediating the beneficial effects of maternal exercise on offspring metabolic health. I've had these pictures up here at the beginning of my talk. And one day I realized I was giving a talk. I said, these are actually my offspring. So I'm talking about offspring health, and here are my offspring. You know, I've been very fortunate. I've got 3 kids. They're all in college now. They've all been they're all healthy, and they're all exercising. It's been a lot of fun to see them grow up, and now they're all going into science. I don't know why, but that's what they're doing. So wasn't my influence at all. So in the lab, as Ann mentioned, we have a lot of different areas of research that have been going on over the years. And I just want to start on these, touch on these before then I talk, and you'll see how these things have all sort of interconnected into the area that, this one area that we're working on now. So we've done a lot of work looking at the effects of single bouts and exercise training on skeletal muscle, a lot of adaptation studies, cell signaling and AMP kinase work. But what we've really come to realize, the whole field has come to realize, is that exercise isn't just about what happens to skeletal muscle, but really probably every single tissue and cell and organ in the body is affected by exercise. And so we've really expanded our research to look at other types of adaptations that occur with exercise. Along with this, we now know that there's very clear intertissue communication, tissue-to-tissue communication that occurs with single bouts of exercise or exercise training. And this is probably incredibly important for all the health benefits of exercise. As Zen also mentioned, we have a large focus on the lab on what happens to adipose tissue when you exercise. I could have given a talk on that. I ended up going with the maternal exercise. We're looking at adaptations that occur where we get changes in adipokines. And we think that this adipose tissue adaptations is really important in the regulation of systemic metabolism that occurs with exercise. And I will actually circle back to this towards the end of my talk. And then we have a big part of the lab is working on the effects of maternal exercise on offspring health. We're also doing work on paternal exercise. I won't talk about that today, but I will talk about these maternal effects of exercise. So how did we get into studying maternal exercise and its effects on offspring health? Well, it really stems from the fact that we know that there's really high rates of obesity throughout the world and all the complications and increases in type 2 diabetes. But what we also know is that there are very high rates of obesity in women of childbearing age. So over 36% of women in this, what we might consider the childbearing age of 20 to 39, that have BMIs of greater than 30. And this is a real problem, not only for the woman that is obese, but we also know through a number of studies now that the offspring as a child has increased susceptibility to be obese. And as that offspring becomes older, there's also an increased risk for obesity and type 2 diabetes, just by virtue of the fact that the mother was obese. And it doesn't seem to end there, because the grandchildren of this mother also have this increased risk. So if the mother is obese during pregnancy, there is increased risk for offspring from subsequent generations to develop obesity and type 2 diabetes, which we think is part of the problem that is fueling the worldwide epidemic of type 2 diabetes. So is there a means to reduce the transmission of increased risk of metabolic disease to offspring? So this is exactly what I was thinking. I was at Joslin. I was listening to one of our seminar speakers that was talking about this. And I thought, okay, well, if obesity is so bad, could exercise maybe overcome this problem? So of course we started to do these studies on exercise. Now, we would love to do these studies in human subjects, but as I said, most of the effects that are seen by the mother being obese occur when the next generation is middle-aged. So it's very difficult to do studies, 40, 50 years in location, in time. And so we have worked using rodent models, as of other people in this field. It's really an in our case, using spontaneous exercise or voluntary exercise in mouse models and then looking at the offspring. So our original hypothesis when we started this work was that increased physical activity of the mothers will improve the metabolic phenotype of the offspring. And so what I'm going to talk about today in terms of the data, first, I'm going to talk about what I consider to be the remarkable effects of maternal exercise on offspring metabolic health, and hopefully I'll convince you that it is quite remarkable what can happen. And then I'll talk about the discovery of an exercise-induced placental protein. These first two parts have been published, so I'll try to go through them fairly briefly and show a lot of cartoons about what we think is happening. And then I'm going to get into more of our recent work on how the exercise moms may be talking to the placenta of the offspring. And so really maternal placental fetal communication. And then finally, I'll just show a slide on the effects of what happens when the grandmothers exercise. So the effects of maternal exercise on offspring metabolic health. This work was started in our lab by Kristen Stanford, who's now faculty at Ohio State University. And so, let's see. And so the model that we use then, and we still continue to use now for the most part, is we have groups of females that typically will have two groups that are chow fed, and they will be housed in standard cages for a period of 2 weeks, or they're housed in these voluntary wheels, so they're the train group. And then we remove them from the cages. They breed for 3 days with the males. The males are not treated. And then they're put back either in their sedentary cages or they're trained where they do exercise. And then we also typically do this under conditions where the moms are also eating the high-fat diet. So we typically will have 4 groups. And do the animals exercise? Yes, they do. They do about 2 to 3 kilometers a day during pregnancy. It's not as much as a non-pregnant animal will do. We'll do more like 6 kilometers, but still doing a significant amount of exercise, and they have training adaptations. And then for all these studies that I'm going to talk about today anyway, the male breeders are sedentary and chow fed. So the treatment is of the females. So here's another way to look at it. So we have our females, our dams are actually, they're trained or before and during the pregnancy, and then they're either in the wheel run or they're sedentary, and then they're plus or minus a high fat diet. And then we're looking at the offspring as they age, but I'll just show you some of the adults. So here is glucose tolerance, a major phenotype that we're always looking at. And this is done, I'm just showing the data from 52 weeks of age. And what you can see, and again, thinking about this, glucose tolerance, the lower the level, the healthier the animal. And you can see that if we compare the offspring, now these are offspring data, this is the glucose tolerance level of an offspring of a mom that was sedentary versus a mom that was trained. So if the mom trained the offspring at a year of age, which is middle age for a mouse, has a clear improvement in glucose tolerance, really quite remarkable. Now, what happens if the mom also ate a high-fat diet? What happens to the offspring? You can see a much worsening of glucose tolerance if the mom ate a high-fat diet. But remarkably, if the mom also was eating the high-fat diet but was also training, the glucose tolerance is completely normal. So very dramatic effects of the high-fat feeding of the mom on the offspring. Again, this is a year later after their middle age, but the mom training, doing this voluntary wheel running completely reversed these bad effects of the high-fat diet and had just even effects under chow-fed conditions. And yeah,
52 weeks for a mouse is actually pretty old.
It's about middle, yeah.
That's pretty old.
Yeah.
So did you look at intermediate ages?
Yeah.
In terms of reproductive fitness, et cetera?
Yeah. So we look as early as 8 weeks. We don't see any real phenotype until sometimes we see it as early as 36 weeks of age, but typically we don't see it until the animals get older. And so these were our initial data that we published, but I should mention that I think this is very clear now. There's been multiple studies, including papers from Zen's lab that have also shown this. that there's clearly effects of the maternal exercise on the offspring health. People have used different types of exercise and also have used rats and see this. So it's a consistent finding. So the next thing that we wanted to do to really understand this is we wanted to understand what were the offspring tissues that were responsible for the improved glucose tolerance in the offspring of the mothers that exercised. And so we looked at skeletal muscle, and then also you looked at skeletal muscle and saw some effects. We didn't see much of an effect on skeletal muscle, but what we did see were changes in the offspring liver. And this is work done by Joji Kusayama, who also now has his own laboratory back in Japan. And most of the work now that I'll talk about is from Joji. And so again, I don't have time to go through all the intricate details of this work, so I'll show you some sort of cartoon types of summary of what he's found. So basically, what we can do is that, all right, so we know that in the mom exercises, and we look at the offspring, we see glucose tolerance, and we see this improvement in glucose tolerance. So we can then take the liver from these offspring at various ages, as early as E13.5 all the way up to a year, and we've studied the livers of these offspring. And so the first thing we wanted to see is, okay, so if you have improved glucose tolerance, is hepatic glucose production decreased under conditions when the mom exercised? And that's exactly what we saw, looking at isolated hepatocyte glucose production and also doing clamps. So definitely glucose production was decreased. Then Joji worked backwards, and he started to look at the whole gene expression profile of the liver, and he saw this improvement in gene expression that we think is important for this change in decreased glucose production. So how is gene expression changing? Again, this is very much cartoon form. This is years worth of work, but what he found was that there was demethylation, so these epigenetic changes marked by demethylation that occurred that resulted in these changes in gene expression. And then, so he figured this out, that there was this demethylation of all these promoters of these important hepatic genes. So that was very exciting to see that these changes were occurring when the moms, in the liver offspring of the moms that exercised. The next question was, well, what's the signal? Well, how does this demethylation epigenetic change occur? So what he did was he figured out through a lot of experiments that what actually happens is when the mom exercises, you get an activation of AMPK in the liver, which then regulates this IBH alpha-ketoglutarate and TET signaling pathway that regulates this demethylation. So again, a lot of experiments, a lot of knockdown, all sorts of different experiments to show that all these components were happening. So the next question then is, we see that there's these remarkable effects on the liver, leading to changes in improved glucose tolerance, but we started to think more clinically in a sense. Okay, so if there are all these changes going on in the liver, we really need to understand how that's happening, because perhaps this could be an eventual treatment for people, for mothers to activate this process happening in the liver in the offspring. And again, a lot of this work was done at day E13.5, where it's the fetal liver that's having these adaptations. So we want to really understand, how does the exercising mother stimulate this AMPK TET signaling, leading to DNA demethylation and this decreased glucose production in the offspring liver. So at this point, I think Joji did a very interesting and important experiment. It was a serum stimulation experiment of primary hepatoblasts. So what he did was he took serum from sedentary pregnant females, and then he took serum from trained pregnant females. And he also took serum from trained females that were not pregnant. So he had the serum, and he's hypothesizing that there's something in the serum of the moms that's regulating the liver. And so then he took primary hepatoblasts just from untreated dams, and he did this serum stimulation experiment. And then he looked at this analysis of this AMPK TET signaling axis to see what would happen. So here on the left-hand side, I have the signaling axis, the AMPK, the IDH, the TET, which we know is there's multiple isoforms, et cetera, but we know is very important in the regulation of this demethylation process that regulates gene expression. So over here, we're looking at TET right now. And what you can see is that this is the control. And if you put on the hepatoblast the serum from the sedentary moms, you don't see any effect on TET expression. This is the TET1 isoform. But if you put the serum from the trained moms on these hepatoblasts, you see a clear increase in this TET protein, suggesting again that the serum is really what's important. But what was really interesting then is when he put the serum from the trained females that were not pregnant, there was no increase in TET1. And this was the case for all the different, the TET proteins and also the IDH proteins, as well as looking at AMPK phosphorylation. You can see it only had this effect on the liver when the serum was from a trained mom, as well as looking at the various genes. And again, he did all the different things. So what this really suggests then is that you can't just train a female and have this serum changing and have this effect on the hepatoblast. You have to also have the mom being pregnant, which means that you really need a placenta. We assume you need a placenta in order to have this effect. So that really helped us in trying to understand then what was happening. So only serum from trained pregnant female activate the AMPK TET signaling pathway, DNA demethylation, and the gene expression. So of course, what we want to find out then is, of course, what is this protein? So we can see that the serum from the trained dam mimics the effect of maternal exercise on offspring. And we want to see what is the serum factor from the trained dams responsible for the activation of the AMPK TET signaling acids. So this led then to the next part, which is the discovery of an exercise-induced placental protein. And this work was, again, spearheaded by Joji, and Anna Wagner from the lab also was very instrumental in this work. Okay, so what was done next was we did in collaboration from the core lab at Harvard, we did mass spec analysis of the dam-derived serum, comparing serum from sedentary dams and trained dams. And what was found was that there were 577 proteins. And then you need to narrow it down, right? So, yeah.
Very interesting phenotype change. My question is, if, let's say, in a human woman, and if they do not get pregnant, but they do exercise, this will not help the later on if they got pregnant. But only when they get pregnant, then they do exercise. Is that true? That means women, if you do exercise, it's nothing to do with your baby later on. But if you do exercise at the same time got pregnant, it's going to help the baby. Is that true?
Well, we don't know exactly the answer, but what I can tell you is that the initial studies that we did, we actually did 3 different types of exercise of the moms. We did just exercising before, we did just exercising during the pregnancy, and we did exercising both before and during the pregnancy. And while there was some effect of if the moms exercise only during the pregnancy, there was the most clear effect when the mom exercised both during the pregnancy, before the pregnancy and during the pregnancy. So I think it's, as you'll see when I'm talking about the proteins that we're studying, it's clearly the placenta is needed for some of these effects, but I think that the exercising prior to the breeding is actually contributing also, and we don't know exactly how. So, okay, so we're trying to understand what's the factor in the serum that's so important. So we need to narrow it down now. So of these 577, Joji thought, okay, I'm going to study proteins that are upregulated. There are probably some things that are important that are going downregulated, but he decided to focus on proteins that were upregulated. So he found that 78 of these proteins were upregulated by 1.5-fold or more in the serum from the trained dams. And then he assumed that these proteins would be secretory proteins, proteins that were secreted. So looking at Go databases, then he narrowed it down to 19 different proteins. And then again, the effect seem to be, placenta needs to be present in order to have these effects on the liver phenotype. And only 4 proteins were known to be expressed in the placenta. And of those 4 proteins, he found that one of these proteins had previously been reported to phosphorylate AMPK. And we knew that this phenotype in the liver AMPK was necessary to have this effect. So he took this 577, he got all the way down to 1 protein. And so what is this protein? Well, this protein is superoxide dismutase 3, SOD3, also referred to as extracellular SOD or EC SOD. It has a classical role as an antioxidant protein, but we discovered at that time it had new roles, had been suggested to have new roles as a signaling protein. So we thought, okay, this is a pretty good candidate here. So the first thing we wanted to do right away, we wanted to see whether exercise training would actually increase SOD3 protein in the serum and the tissues of the dams. Yeah.
Just curious, in the literature, SOD3 phosphorylate AMPK, how does that happen? This is superoxide scavenger, and how does that phosphorylate AMPK?
We don't know.
Okay.
It's not known, and we're still trying to figure that out. Yeah.
In the presence of SOD3 in serum, AMPK gets phosphorylated, or is there a direct enzymatic effect of SOD3? So if you take SOD3, AMPK, and phosphate, you get phosphorylation.
No.
Okay, so
No,
it's indirect.
It's indirect, yes, it's indirect. Okay, so does SOD3, if this is our hypothesis, does SOD3 actually go up? Can we measure this? And so this is data from the dams. This is not the offspring. So here's serum SOD3. And so the moms that were sedentary trained under chow or sedentary trained under high fat. And this is at day E13.5 of pregnancy. And you can see that, yes, when the moms train, the SOD3 does increase. But if you take just a female that's not pregnant, who's trained, doing the same training, and you'll see that there's no increase. So again, suggesting that the placenta is key for this increase in this very important protein SOD3. So then what happens to the various tissues in the mom? Like, where is the SOD3 coming from? Again, we're assuming it's coming from the placenta, but we don't really know. So this is just these are all pregnant, trained, untrained, the 4 different treatment groups. And what you can see is we don't see any changes in the expression of SOD3. We actually see a little bit of a decrease in the subcutaneous white adipose tissue, but none of the other tissues that we looked at, we don't see any change in SOD3, except when we look at the placenta and we look at the trophoblast, and you can see very clear increases in SOD3. So again, suggesting it's the trophoblasts probably of the placenta where this SOD3 is coming from. So then SOD3 is increased in the serum of trained pregnant dams, and exercise training increased SOD3 mRNA and protein only in the placenta and the trophoblast. So this really suggested to us then that SOD3 may be a critical placental protein that plays a fundamental role in fetal development, offspring health and adulthood, and maternal health. And so we started to really investigate SOD3 more. Yeah.
Is SOD3 increased in baby blood stream?
Is it what?
It's increased in baby's blood stream.
Yes, we can see an increase in the fetal, in the fetal. It was very hard to measure, but yes, we can see it in there too. Okay, so if SOD3 is so important, can SOD3, if you just take recombinant SOD3, can you mimic these beneficial effects of the effects of exercise that we see on the epigenetic changes?
Quick question, does
Yeah.
SOD3 pass the placental barrier?
We are assuming that it is, you mean going out into we're assuming that, I'll actually have a little.
In other words, does it get to the fetus?
Yes, we believe that it gets to the fetus. It's in the fetal serum. And we think it goes in the maternal artery out to the mother. So it's in the placenta, which is part of the fetus, and then, yep. Okay, so we looked at, we used, again, we went back to primary hepatoblasts and we treated them with SOD3, and then we looked at all the signaling. Again, I'm not going to show you all the data, but you can see very clearly when you treat with SOD3, this is a time course, this is a physiological concentration based on the measurements that we made in the serum. You can see clearly that this SOD3 is increasing the phospho-AMPK. Again, we don't know the exact mechanism for this. And if we look at the expression of these IDH and TET proteins, they're all being increased by SOD3, as well as looking at the expression of these genes that we see go up with a maternal exercise. So what this then suggests is that when the mom exercises, the placenta is increasing the expression of SOD3. Again, we believe that this is then going to the fetal liver to affect the fetal liver, and also it's showing up in the maternal artery and showing up in the mother's serum as well. Within the fetus, then you'll see that the liver, that the SOD3 then we believe is activating and having all these effects on the liver. So our hypothesis is that placental-derived SOD3 mediates the benefits of maternal exercise on offspring health. So to really try to get, though, at what's happening, yeah.
So there's a question online. So
Okay.
what might this mean for the offspring of the people like me? This person said, like me. She was pregnant, who were much less active during pregnancy compared to due to her fatigue and nausea. So what it means to men for her offspring?
Well, that's a very good question. We don't know, right? I mean, we can make some assumptions, but well, I'll wait to the end and I'll answer that question, because then I have a funny story about myself. So I will.
I have a question.
Yeah.
That is that if this is impacted during pregnancy,
Yeah.
that means the impact of SOD3 and the AMPK is during the embryonic development of the liver that impact the future, because the baby, it's unlikely that the SOD is going to stay there forever until they are 5 months old.
Right, right. Yes, so we think that the peak effect of this is occurring at about in the female, probably in the second trimester, and well, in both in the mouse and the female. We think that is probably second and third trimester based on some time course studies that we did. Yeah. Okay, so if we really want to understand what's happening, we really need to we need to knock out SOD3. And so we were in collaboration, we were able to do this. So we did a placental specific SOD3 knockout, as you can see here. Again, here in the floxed animals, you see the increase in SOD3 in the placenta. This is the protein, obviously. And then this is gone in the knockout. And so then we can use these animals, and it's actually a nice setup because your maternal treatment is the same, because you'll have some wild type and some knockout placenta. So if we knock out then SOD3, what happens to this whole signaling pathway? And we see that these are all downregulated. You don't see the effects of exercise. Again, I'm sorry, I'm showing you this by cartoon. I don't have time to show you all the individual data. But what I will show you, and I think what's really important is, okay, so you're getting rid of the SOD3 in the placenta, then what's happening to the offspring glucose tolerance, right? This is really the key question. We want to know what the phenotype is going to be. So, so we've done GTTs and serum. We've actually done lots of things, but GTT and serum insulin in offspring at 52 weeks of age. We've done the whole time course, but what I'll show you here now is offspring glucose tolerance. So here's the floxed animals. So if the mom exercised, you can see that there is a training effect, as we had seen before. But in the absence of the SOD3 in the placenta, you see not only a lack of exercise effect, but a worsening of glucose tolerance. And if you look at the offspring serum insulin concentrations, and also very important phenotype, we also see that insulin concentrations are lower if the mom exercised in the trained offspring, and a worsening if there was no SOD3.
There a heterozygote effect?
So we haven't really done a head for this type of experiment. So it really shows that placental-derived SOD3 mediates the beneficial effects of maternal exercise on offspring metabolic health. So you get rid of the SOD3 in the placenta, and then the offspring have a worsening glucose tolerance. Yeah.
The offspring, are they always maintaining sedentary chow?
Yes. Yeah.
Like, yeah, so I guess my question is thinking about like, why would this mechanism exist, right? To prepare an offspring, and a lot of times we think about it, right? To prepare an offspring for the environment they're going to encounter.
Mhm.
And I was wondering if you had thought, or if it kind of fits in this model of matching the environment that the offspring encounters to the environment that the mother was in during pregnancy, right?
Yeah.
So if they're always chow and sedentary, that's a different environment than they were prepared for in the womb.
Right. Yeah, that's an interesting way to look at it, right? They weren't prepared for sitting around and doing nothing. They were prepared. We've actually done some studies where we've put the offspring in wheels and see how they adapt. They don't seem that different, regardless of whether the mom exercised or not. So I mean, it's not exactly answering your question, but we have tried to look at that. Like if the mom exercised, do the offspring exercise more than, or is their metabolic phenotype different? If you compare offspring that were put in cages or not put in wheel cages as they're developing, do the offspring of the moms that had exercised, have a better metabolic response? And that we really don't see a big difference, or we don't really see a difference. I think that another way to look at this, and I should have mentioned this before, is we think of the treatment as being the exercise for the moms, right? But in the wild, the moms are doing exercise normally. So the treatment is really, of the moms, is really more not having the wheels. So
Their condition is weird for a mouse.
right. They're just sitting in a cage by themselves as opposed to, so I think that's another way to try to look at that a little bit. So, okay, so placental-derived SOD3 mediates the beneficial effects of maternal exercise. Again, this is what we think is that SOD3 is an important, but then how does the exercise actually increase the placental SOD3? And if we think about it, we know from work, a lot of work in the exercise field right now, is that when an animal exercises, probably multiple tissues, not just muscle, but multiple tissues are secreting factors, right? We know these myokines, adipokines, all different kinds that are being secreted. And so our hypothesis is that when the mom exercises, that she's secreting these exercise transducing molecules that then are probably affecting the trophoblasts of the placenta, which then are regulating transcription factors that then may in turn increase SOD3. So I'm going to go through this pretty quickly. So basically what we did was we did RNA-seq of placenta, of sedentary placenta and trained placenta, and looked at transcription factors. These are all the transcription factors that were increased. And the one I'm going to talk about is VDR. This is the vitamin D receptor. It went up 4-fold and was highly significant. And we show that vitamin D receptor actually does increase SOD3 expression. So training increased placental vitamin D receptor. So we thought, aha, this is really interesting, right? You're getting not only an increase in SOD3, but you're also getting this increase in VDR. We confirmed this with actual experiments of placenta, not just the RNA-seq. And so we thought, so vitamin D may be really important here, right? This is the VDR. So we could do these experiments. So we could ask, is vitamin D necessary for the effects of maternal exercise to improve the offspring health? And so we can do that by feeding the animals a low vitamin D diet to see if this is necessary for the effects. And we can also determine whether vitamin D treatment functions as an exercise mimetic and increase placental SOD3 and improve offspring metabolic health by feeding in animals high vitamin D. And we're pretty excited about this, right? Because we're thinking, aha, moms don't have to go out and run marathons or anything like that. Maybe they can just take high vitamin D and have these beneficial effects of exercise if it's going through the VDR. So this is what happens. So when you do these experiments with the low and high vitamin D, and you look at the offspring health a year later. So here is the normal vitamin D diet. This is the improvement in glucose tolerance that we see of the mom trained. If the moms are fed a low vitamin D diet, you lose this beneficial effects of training, and you actually have a worsening of glucose tolerance. So it is important. Vitamin D is really important. You have to have the vitamin D in order for the animals to have a normal phenotype. But then what happens if you just give the mom high levels of vitamin D? So this is shown here. If you compare this bar with this bar, if you just give the animal high vitamin D, you do not have an improvement in glucose tolerance. However, if you have the high vitamin D and the training, you actually get a little bit of an augmentation of the effect. But you can't just take vitamin D and mimic these effects of exercise training. Yeah.
Endogenous vitamin D level in your training.
How?
The endogenous vitamin D.
Oh, yeah, that's a good question. And we're trying to do those studies in humans right now. There aren't major effects of training, as far as I know, on vitamin D. Yeah, so we are trying to understand in studies done in human, I was going to mention this at the end, in human studies that we're doing to see whether the vitamin D status of the moms in correlation with the exercise are affecting the offspring. Okay, so normal vitamin D intake is necessary for maternal exercise to improve offspring metabolic health, and vitamin D treatment alone does not function as an exercise mimetic to improve offspring metabolic health. So placental-derived SOD3 and vitamin D mediate the beneficial effects of maternal exercise on offspring health. Yep.
So in regions of the globe where winter pregnancies are also dark pregnancies, completely dark, and there is a higher probability of vitamin D deficiency, is there?
Yeah, that's what we are now doing collaborations
Would you
with,
expect epidemiological?
right, that there may be some issues going on there. So it really, I think,
Could the mice
increase their,
increase their UV exposure or decrease?
no, because, no, we haven't done that. But now we're wondering, the important role, as you're saying, of sunshine. I mean, this has potential really important implications for human health, right? So those are the types of things we're trying to understand right now. Yeah.
There's an online question whether this exercise effect is exercise effect alone or associated with the BMI. I think that's a human research question.
Yeah, so we haven't really done that type of study in humans. We haven't seen an association. I'm trying to remember, because we did a big cohort, and I'll show you this data, not with BMI, but I don't think we see any association between BMI and the like SOD3 levels or those types of things. So, so I would say no, we didn't see that.
Maybe the question is that impact has to be in the high BMI group with exercise will show impact, because the lean pregnancy may not have much of a show, right?
Yep, it's possible. Although at least in the mouse models, we see the effects in the lean chow-fed animals. We see it in the obese, but we also see it in the lean. Yeah.
I have a question that might also be a human subjects research question, but have you any idea of dosage of exercise?
Yeah.
You said marathons earlier, and I was thinking about the translation of this for humans.
Yeah, so I get that question every time I give this talk, and it's important. So we decided when we put in our renewal for this grant, I said, okay, everybody asked this question, we're going to do this. And the reviewers hated that we were going to do that. But we are still trying to do it. And we're trying to understand, at least in the mouse model, what exercise intensity do you need in order to have these effects? What we've learned is it's very difficult to measure VO2 in pregnant females. So we're using metabolic cages to measure VO2 max and exercise. So then we're trying to adjust. We're trying to measure VO2 max, and we have in pregnant females, and then adjust the intensity of the exercise that they're doing by doing treadmill training It's a very difficult experiment, but hopefully we'll have an understanding. I mean, everything we did up until this point where we're trying to do the treadmill, at least in our lab, has been voluntary exercise. So we can't translate exactly what that means in humans. But I will show you one slide on human data comparing women that basically get no exercise to some exercise, and you'll see that in terms of SOD3, it is affecting it. So anyway.
Are all these benefits related to aerobic training? Is there anything associated with resistance training? Or
Yeah.
stretching?
Yeah, that's another thing that we would like to do, would be to do resistance training to see whether these effects are also occurring if a mouse does resistance training. I mean, mouse resistance training is not easy to do, which is one of the reasons that we haven't done it, but we would also like to do that too. So, okay, so I have quite a bit more to do. So I will try to get to you some of the newer data. All right, so maternal exercise. Okay, so I just want to remind everybody that, and I told Zen I was going to show this one slide on maternal behavior. So SOD3 is going up in the moms, right? This is shown here. And so we thought, well, could this SOD3 actually be affecting the mother's behavior? So do SOD3 levels in the serum of mother affect maternal behavior? So in collaboration with Joji, who's back in Japan, we've done these experiments, and I won't go into, it's very complicated making these animals, but so we have a wild type where the placentae are all flocks, so they're all wild type, and then we have a het, and then we have a knockout where all the placenta are SOD3 negative. And how does this SOD3 in the placenta affect this maternal serum? And you can see that in the knockout, you get a decrease in the maternal serum SOD3. So serum SOD3, we've successfully gotten the serum SOD3 in the mom's decrease. So how does this affect the way she behaves? And it's really quite interesting. So there are all these measurements that you can do for maternal nurturing behavior. So you can look how many pups actually have milk in their stomachs. And you can see this is significantly decreased. This is a percentage of the pups with milk in the knockouts. The pups that are actually stay in the nest, these is decreased if the mom has low SOD3 levels. The cleaning of the pups, another measurement, is decreased, as well as the number of living pups at postnatal day 10 is really dramatically decreased. So I think these are really striking behaviors of the mom if the SOD3 levels are lower. And so now we're really interested in understanding how the maternal SOD3 is affecting the mother's brain and her behavior.
Pups are not SOD3.
They're not, yeah, it's the mom. So, and then, yeah, so there's also, you can also look to see if the pups of the placenta doesn't have and you can compare that, and there's no effect of the pups. It's the mom's SOD. So placental SOD3 and vitamin D mediate the beneficial effects of maternal exercise on offspring metabolic health, and then the placental-derived SOD3 mediates maternal nurturing behavior. So how does the maternal exercise transmit a signal to the placenta? So this is another really important question, and this is this maternal placental fetal communication. And this is work from Joji, but also from Shivari, a postdoc in the lab, and Madison, our research assistant in the lab, who've done this work. So how does this happen? How do we get this effect on the trophoblast? Our hypothesis is that there's these exercise transducing molecules that then regulate the VDR, that then regulate the SOD3. So what could these exercise transducing molecules be that are coming from the maternal tissue? So again, this goes back to what I talked about at the beginning. We know that there's lots of intertissue communication that goes on, and we've also done a lot of work on adipokines. And one of the studies that we published a year or so ago showed that when you train a mouse, this is not a pregnancy study, this is just a training study, you get an increase in TGF-beta2. Again, I'm summarizing years worth of work, and that this TGF-beta2 actually can go into the circulation and can have really important effects on other tissues in the body that can result in improved systemic metabolism. So we decided, let's check out TGF-beta2. Maybe this is important in the maternal exercise. Maybe the mom is making more TGF-beta2 that's having some effects. So basically what we're saying is that could this TGF-beta2 coming from the mom's adipose tissue then be talking to the trophoblast in the placenta? And so the first thing then is, okay, well, if you just take TGF-beta2, does it affect the VDR expression, right? Because if this doesn't happen, then it's probably not that important. So did the experiment, just took trophoblast cells, treated them with TGF-beta2 at physiological concentrations that we know from all our other studies. And sure enough, the TGF-beta2 significantly increases the VDR. So we thought, okay, this is worth looking at a little bit more. And so our hypothesis then was that the exercising maternal adipose tissue, TGF-beta, could transmit a signal to the trophoblast cells of the placenta. And so then does the exercise in the pregnant mothers increase TGF-beta2 in the adipose tissue in the dam serum? So we need to see that this is happening too. Now here's all the different tissues. This is looking at TGF-beta2, and you can see that certainly just as we had seen in a non-pregnant animal, TGF-beta2 goes up in the adipose tissue, not in other tissues. And also, if you look at TGF-beta2 concentrations in the serum of the pregnant dam, that they go up. So then what we need to do then is we have these adipose tissue specific TGF-beta2 knockouts. And so we took these animals and then we looked to see how this lack of TGF-beta2 in the mom's adipose tissue would affect her and then affect the offspring. So we're partway through these studies. We haven't finished these studies, but you can see that again, in these, the maternal serum, the TGF-beta2 goes up when the mom exercises, and this effect is lost if the mom doesn't have TGF-beta2 in her adipose tissue. So then what happens in the fetus? So, and what we see is that here's the fetal serum SOD3, and here again, this is the increase that we see of the mom exercise. And you can see it's not fully inhibited, but it is definitely partially inhibited, this increase in SOD3. And if we look at the VDR expression in the placenta, again, this is inhibited if the mom doesn't have this TGF-beta2 coming from the adipose tissue. And we've looked at all the different promoters of the different genes that we think are important in the liver. And again, there's clearly an effect. What we don't have data on yet, but it's looking pretty promising so far, is that if the mom doesn't have any TGF-beta2 in her adipose tissue, the offspring don't have as clear an improvement in glucose tolerance. So we think that TGF-beta2 is actually really critical for this effect. So if you knock out the TGF-beta2, you have effects on VDR and SOD3. And again, we're really looking at the glucose tolerance now of these offspring. We have to let them age up before we can do these studies. Okay, so I talked a lot about the animal work and a lot of these mechanisms, but of course, as many of you have asked the questions, is this relevant to human physiology, right? Because it could have very clear clinical implications. So we've done some different collaborations, one with the University of Ottawa, and what we did was we compared, or what they did, and they provided us samples, is we compared groups of women that were either inactive, they only did 2 minutes of exercise, moderate vigorous physical exercise a day, or they did as much as 40, they averaged 40. So big difference. So then what happens to their SOD3? So in the moms that we're exercising, this is in the second trimester, you can see the physically active women have significantly higher SOD3. And if you look at the placental SOD3 at birth, you can see this is also significantly increased. So at least in terms of SOD3, it looks like consistent with what's happening in the animal models, the SOD3 is increasing when the moms are more active. And TGF-beta2, which we talked about in terms of the adipose tissue, what happens there? And if we look at TGF-beta2 concentrations in the more active women, we can see that it's higher than the inactive women. So consistent with this hypothesis of TGF-beta2 and SOD3. So just to summarize then, we see that SOD3 is increased in the serum and placenta from physically active pregnant mice and women. Placental SOD3 and vitamin D are required for the benefits of maternal exercise on offspring metabolic health. This is animal studies. Placental SOD3 regulates maternal behavior, so we think it's very important. Of course, we have no idea in humans, but in the mouse models, this looks pretty clear. And then the mechanism, lots of different things going on that are having these beneficial effects, but we think that exercise increases maternal adipose tissue TGF-beta2, and it's a potential mechanism for this improvement in offspring health. So just I'll show one slide, or maybe two, just the work of Anna Wagner. I think this is really quite remarkable work. What happens when the grandmother exercises on the F2 offspring? And so our hypothesis was that increased physical activity of the grandmothers will improve the metabolic phenotype now of the F2 offspring. So there's no treatment of the F1. It's just the grandmothers that are exercising and what happens to the F2. These are from male F1. We now have data from female F1 generating the F2 as well, and we see the same thing. So here, this is now an aging effect of the age of offspring as they're getting older. You can see if the grandmas were sedentary, you get a worsening of glucose tolerance as an animal ages. This is male offspring. We see the same thing in female offspring. This is typical of what we get. But what happens if the grandmother exercise? Again, the F1s were treated with just chow fed and were just normally, and you can see really, really remarkable effects. If the grandmothers exercise, you get this huge improvement in glucose tolerance in the grandpups. And if you look at HOMA-IR, a measurement of insulin sensitivity, you also see this effect. And I should say that body weight of the offspring is similar. Out at about 52 weeks of age, you start to see a separation of the body weight, but even at ages where you see improvement in glucose tolerance, there's no effects. So maternal exercise improves the metabolic health of both first and second generation offspring. There's lots and lots that we need to do. We need to really, again, we're starting to look at TGF-beta2, but we think there are probably other factors that are regulating the placenta. These proteins are probably extremely important. We also want to understand the placental secretome and transcriptome, which we have those studies going on right now. We also want to understand, we focused on the liver, but we now have a lot of work going on looking at other tissues, especially the brain, to see what's happening in the brain in these offspring, and also looking at the sperm and the oocyte to understand epigenetic regulation, doing a lot of small RNA analysis with a group from UMass, and also looking at the F2 in more detail. As I mentioned before, and as we talked about, in terms of human studies, we want to understand the role of vitamin D in pregnant women. What's the optimal maternal exercise paradigm for maximal health benefits? Again, another, so you all had all these questions. And then how does maternal exercise alter the placental secretome? So finally, should women exercise during pregnancy? I'm not an obstetrician, but I would say that being physically active is probably a very important thing. Maternal exercise, at least the animal studies show, is probably a means to reduce the transmission of increased risk of metabolic disease to offspring. And I think this is a great example where we determine the physiological and epigenetic mechanisms mediating the benefits of maternal exercise on offspring can lead to new therapies to prevent transmission of metabolic disease. So again, very important to, I think, to do these basic science type studies, physiological types of studies, to lead us to really understanding how can we prevent all of this metabolic disease that's going on. So finally, just acknowledge the funding from NIDDK and fellow support from ADA and other places. This is our lab group, not this year, the year before. We even have Santa Claus working on our side. But it's a great group. This is Sarah's lab, who, as you heard, is coming, and my lab together. I tried to mention everybody there. Mike Hirschman is our lab manager, who really runs everything. And then past members of the group who have contributed to this work. And then lots of collaborators at Joslin and at different institutions, who I've tried to measure. Okay, so I'll stop there. And I don't know if there's time for questions, but be happy to answer some if there are more questions.
Yeah, before answer some of the questions, there are two online questions. One is that exercise mice eat more. Is that there is a possibility they eat more and they have more consumption of vitamin D? And for I think the babies they recommend to use vitamin D now. There's a question about whether that alone is sufficient. I think you answered that question.
Yeah.
And then the second question is about a circadian rhythm, when to exercise to have the best benefit.
Okay, so in terms of the circadian rhythms, we don't know. I mean, we do know when they exercise, but we don't, to try to translate that, I don't think we can tell that from the mouse because they're doing voluntary exercise. They exercise when they want to, presumably they're doing it during their dark cycle. We don't really see any activity during the daytime. So, but that's another very important, probably could be a very important question for clinical. And the very first question, I can't remember what that was.
Whether vitamin D due to mouse consumption when they
food
exercise, they
consumption,
eat
yeah.
more and
Yeah, I don't know. That's an interesting idea. We should try to do the calculation. They don't eat that much more. The females seem to maintain their body weight pretty well. So their food intake is if you take a sedentary pregnant mom versus a trained pregnant mom, I don't think their food intake is not dramatically different, but.
the impact is restricted to that period of embryonic development. That is an important question.
Yeah, so we find that in the mice and even in the human studies where we measured SOD3 in the serum of moms at first, second, and third trimester, we see the greatest effect on the second trimester, and the same thing in the mice. We see that we see the greatest increase in expression in the placenta and the serum in the second trimester of the mice.
So just kind of a follow-up with one of the questions, and I'm kind of curious about your thoughts on this too, is we actually have a collaborator back in Minnesota who was looking at time restricted eating and found that exercise in the evening actually helped with maintain insulin sensitivity throughout the day. So I'm wondering if there's some correlation to the question that was asked online, if there was some type of interaction there. But going back to my original question that I was going to ask was, have you looked at like vitamin D stores? Like, I'm pretty naive about this, but I only know about this because when we look at the microscope and we look at primary hepatocytes, any vitamin that the hepatocyte has is going to be autofluorescent, crazy.
Mhm.
So I'm wondering, is there any correlation from the liver phenotype that you're seeing to like vitamin D stores? There's some type of regulation that's going on in the liver that's also affecting what you're seeing?
We haven't looked at that. We haven't looked at the vitamin D stores at all. That's another interesting idea to try to understand how they're affected. I'm not an expert on vitamin D. I got invited to the Keystone Vitamin D meeting. I was very intimidated to have to talk about this, and I tried to study up on vitamin D a little bit, but I don't know how these diets, because they're commercially available diets, so we should look into that and see what the storage, how it dramatically affects the storage.
And then follow up, have you actually looked at the overlay of the marathon mice with your exercise mothers? If there's like an overlap of the gene expression pattern and methylation markers to actually like that mimic like the marathon mice versus what you see in your exercise induced?
No, we haven't done that. That would be interesting too. Yeah.
This is a very interesting study, great translational potential. My question is, the main key point is the demethylation and also expression of SOD3 in mother and baby. I wonder if there any possibility or maybe any drug, you can just let women take the drug without any exercise, and the baby will be healthy.
Yeah. Yeah, we were hoping we could just take vitamin D and that was to do the trick, and it didn't. Yeah, I mean, and that's that's kind of a goal, right? Is to figure out if there's a way. We still have to understand how the AMPK whole signaling pathway gets activated, and it would have to obviously be something that would go from the mother to the placenta to maybe it doesn't have to be through the placenta, but it would be nice to be able to figure out a way to do that, because not every woman can exercise, right? I mean, so
Kevin and Anthony, yeah.
Thanks, Lori. That's a really nice talk. So I have a bunch of questions. I'm gonna kind of disguise them as two, though. Is the number, mass, or body composition of the pups from the exercising moms different from the sedentary?
Yeah, so, not early on. And not until they're about a year old, you start to see some differences. They're not really pronounced. So, so you can see these effects in terms of glucose tolerance and metabolism at about 36 to 42 weeks at a time when you don't see body weight changes. So there are later, but we don't know if that's secondary to the changes in glucose tolerance. We don't see differences in feeding behavior of the offspring. So we don't really know exactly what the mechanism is.
Are the liver sizes similar?
Litter sizes are similar with the exception of if the moms eat a high-fat diet, litter sizes are a little bit lower. So we cull the animals to make sure everything is the same, but yeah, high-fat diet is pretty consistently seeing that. And I'm saying pretty consistently, you know, other groups too, you see lower litter sizes.
Just real quick, is there any evidence for beiging or browning of adipose tissue in the
Yeah.
offspring from exercise?
We've looked a little bit at that because we study that in the lab, and we've also looked at brown fat, and we don't see any real big differences. One of our hypotheses was that, oh, this is all being mediated by browning or changes in brown fat, and we don't really see much of anything.
Last question.
So the F2 effects, do the F1 mice, if you block the liver effects that are driving the glucose, improved glucose tolerance, do you still get the F2 effects?
Yeah, so what we would like to do is to do some of these experiments in like our SOD3 knockouts, or maybe even some other type of knockout, but probably SOD3 knockout, and see if we lose the effects on the second generation.
The other thing, this is just a little bit of a flyer, but if you foster the pups so that they're not nursed by mothers with exercise,
Yeah.
what happens?
Yeah, so we haven't done those experiments. I think Kristen Stanford has done some of those experiments, and she still sees an effect. But I think that we actually published a paper with her on the changes in the breast milk. So I think that probably is contributing to the overall phenotype, but it's not the whole story.
Great. The take-home message: exercise is better than medicine. No drug can do this to the grandchildren. A good thing to the grandchildren. Thank you.
Well, I'll just say one more thing. You know, somebody asked about what about women who didn't exercise during their pregnancy, right?
Yeah.
So I have I show you my kids at the beginning. So I have twins. Okay, so I was put on bed rest at like 14 weeks. Okay, that's bad, right? Bed rest. And and then we started doing this research, and I thought, oh no, this is really, like, they are doomed, right? So then we did studies with males, and actually, if the dads exercise, you have this same phenotype as you do in the females, which is a whole nother line of our work. So I thought, at least my husband exercises. Maybe they're not doomed. So, but I do not think that bed rest is a good thing for the metabolic health of your. But hopefully at least one of you exercise. So
Thank you so much for such an inspirational speaker. Thank you.